The pesticide chlorpyrifos is a common pesticide used on plants to combat pest and insect invasions. It is also moderately toxic to humans and can cause autoimmune deficiencies. As a result, it is banned in residential areas in many countries. Chlorpyrifos works by inhibiting acetylcholinesterase, which is an enzyme that breaks down acetylcholine. This makes it an effective pesticide.
Chlorpyrifos works by inhibiting acetylcholinesterase, which is an enzyme that breaks down acetylcholine. Acetylcholine is released as a messenger between neurons. Typically, acetylcholinesterase works to hydrolyze acetylcholine in order to break it up into parts that the presynaptic neuron can take up. Acetylcholinesterase allows signal transmission to terminate between a neuron and receptors because there is no longer anything to stimulate the receptors. Without it, muscles are constantly contracted, which causes paralysis.
Bees are very important for plant life because in order to reproduce, plants must get pollen from the stamen, the male sex organs of a plant, to the pistil, the female sex organs of another plant. In order to do so, plants often use bees. When bees go for the sweet nectar found in flowers, they end up getting the pollen stuck on their legs. When the bees fly to another plant, they transfer this pollen to the pistil of the other plant. The sperm cells then fertilize the egg in the plant, and this becomes a seed, which eventually grows into a new plant. Pollination is necessary for the sexual reproduction of plants, and bees are a very important pollinator.
It has been brought to the attention of the scientific community that the agricultural pesticide chlorpyrifos passes off readily as vapor, leading its coverage to a wider scope than intended. It has profound effects on non-target organisms, in particular bee pollinators, as they are in close association with plants. Bees were subjected to varying amounts of chlorpyrifos based on levels in New Zealand in a laboratory setting in order to determine behavioral effects.
Honeybees were collected from 17 sites in a region of New Zealand. The sampling sites were selected to represent a range of land types and agricultural activities such as dry grasslands, riverbeds, and land for grazing livestock. For the behavioral experiment, bees were collected and fed with chlorpyrifos. Although chlorpyrifos is usually present at higher levels in pollen than in nectar, the researchers chose to deliver the pesticide in a sucrose solution because this allowed them to control the exposure more accurately. They fed the honeybees similar levels of chlorpyrifos as had been found in nature. They then put the bees through learning performance tests. During this process, they also manipulated the amount of pesticide to find the threshold value. Some behavioral tests included stimulating with odors by delivering odor-filled air to the antennae of the bees. They rewarded bees when they responded to a certain odor and observed if they responded again in order to show learning. Bees that showed learning extended their mouth towards a certain odor. Another test the researchers conducted was whether the pesticide affected the bees' motor function by seeing if their sensitivity to electric shock was altered.
It was found that chlorpyrifos can negatively affect the learning and memory of bees when present in over 50 picograms. Figures 1 and 2 show that chlorpyrifos decreases appetitive learning and interferes with olfactory memory in both summer and winter bees. Bees treated with chlorpyrifos were less likely and slower to learn to associate an odor with a sucrose reward, and these bees were more likely to incorrectly generalize learning to a dissimilar stimulus odor 1 hour later. Higher doses of chlorpyrifos impaired learning more. However, chlorpyrifos does not negatively affect aversive learning in bees, as is demonstrated by the bees' responses to negative stimuli displayed in Figure 4. These effects may adversely affect the ability of bees exposed to chlorpyrifos to forage and pollinate, especially because olfactory memory is an important part of how bees locate flowers. It is also concerning that bees may be less motivated to seek sucrose when exposed to chlorpyrifos. The threshold dose of chlorpyrifos that affects bees is one that is sometimes present at an over 500% more concentrated rate in many wild bees in agricultural and non-agricultural lands. The effects of chlorpyrifos on bees may therefore be dangerously widespread. Other studies have suggested that field use of insecticides can negatively affect honey and bumblebee populations even if they're not exposed to the field in question until 6 days after insecticide application. Other pesticides that harm bee populations include neonicotinoids such as atrazine and glyphosate, which, like chlorpyrifos, also affect acetylcholinesterase dysregulation and are linked to bee mortality. These findings have important implications for the regulation of chlorpyrifos. Current regulations do not take into account the lasting risk of exposure up to a week after plants have been treated with chlorpyrifos and the disposal of chlorpyrifos to other non-treated areas. However, it should be noted that this lack of regulation is not the case in all countries. Chlorpyrifos have been banned in several countries due to these and similar concerns. This study suggests that the U.S. should perhaps follow suit.
Created using Powtoon.